Welcome to Prospective Doctors MCAT Podcast, brought to you by Med School Coach. Each week, Sam breaks down the highest yield MCAT topics so you can score as high as possible on test day.
Hello and welcome to Prospective Doctors MCAT Basics with your host Sam Smith. The goal of this podcast is to cover the highest yield topics on the MCAT and provide you with some sort of insight into the questions that the MCAT really likes to ask. This podcast is going to cover DNA replication and central dogma. What I'm going to do is I'm going to first go over replication, talk about a few different things there. Then I'll talk about transcription and translation. And then lastly, I'll talk a bit about protein processing. This material is going to show up in 2 out of the 4 MCAT sections. That is the biochem section and also the chem-phys section. And central dogma is a pretty important topic for the MCAT. Obviously, these concepts underlie a lot of the topics that I talked about in the genetics podcast. So I hope this kind of adds to your knowledge on genetics and that this helps you study for the MCAT. So let's get into it. All right, so the first thing I want to talk about here is DNA replication. So DNA replication is the process of duplicating DNA during cell division that occurs within the nucleus of the cell. And this process has been extrapolated to molecular biology, right? PCR is essentially DNA replication, but done in order to amplify some gene of interest or any type of genetic material that you want to look at. And so it's important to understand why cells undergo DNA replication because, it's not like they're just hanging out saying, I got nothing to do, let me just replicate my DNA. DNA replication occurs during mitotic cell division because each of these daughter cells that splits from this parental cell during cell division needs to have a full double-stranded DNA genome. If a cell was unable to replicate its DNA, then during cell division, one of the daughter cells would not get any DNA, and then it couldn't create proteins, and the cell would essentially die. It'd just be useless. So, replication is important during cell division to make sure that each daughter cell gets a full genome. And so, DNA replication occurs during the S phase of the cell cycle. And go listen to my genetics podcast if you're curious about the cell cycle, but just know that DNA replication occurs during the S phase. And before I get into how replication works, I want to talk about one last thing, and that is the difference between semi-conservative, conservative, and dispersive replication. And to me, this is kind of dumb. I've seen it come up on the MCAT a few times, but I think it's kind of dumb because we know exactly how replication works in DNA. Why should we care about these other ways that people theorized DNA replication might work back in the day? Seems kind of silly, but I'm going to go through it anyways because I've seen it. So let's start with dispersive replication. So if replication was dispersive, that would mean that you would start with these 2 parental strands of DNA from a single cell. And then as this cell starts to replicate its DNA in order to be able to split, then you would have dispersion of new bits of DNA and old bits of DNA to create these 2 strands that are now kind of just a mishmash of DNA from the parent and DNA that's newly replicated. So you just get these new strands of DNA that are essentially random bits of new mixed with old, mixed with new, kind of just spread out everywhere. The next and also incorrect model of DNA replication is conservative replication. And so in this case, the parental molecule would essentially direct synthesis of an entirely new double-stranded DNA molecule. In other words, in this case, you would start with a double-stranded parental molecule of DNA, and then at the end of replication, you would still have that molecule intact, but you would also have a brand new double-stranded molecule of DNA. So in a way, conservative replication is kind of like segregating the old and the new DNA from each other, right? One cell would get this parental DNA, the other cell would get this daughter DNA, and there'd be no mixing of that. However, we know that dispersive and conservative replication don't actually exist in cells. Instead, you have semi-conservative replication. Semi-conservative replication is when 2 parental strands separate, and then they each make a copy of themselves. And then so you have these 2 strands of double-stranded DNA, and they each contain 1 strand of new DNA, 1 strand of old DNA. So it's basically like a hybrid. And so we know now that each of these parental strands acts as a template for the new strands. In this way, the DNA polymerase knows exactly what bases to add in as it's replicating the DNA. And so with that, let's get into the 3 different steps of DNA replication. All right, so there are 3 different steps of DNA replication. The first is initiation, the second is elongation, and the third is termination. And I'm going to sound like a broken record because this is kind of the main steps for transcription, translation, and replication. But yeah, just note that they break down pretty easily into those 3 steps. All right, so let's start with initiation. So double-stranded DNA in general is very stable, right? It forms these hydrogen bonds between bases, 3 hydrogen bonds for G's and C's, 2 hydrogen bonds for A's and T's. So it's got to be pried open in order for the replication machinery to get in there and to be able to start replicating these DNA strands. And so how is the cell able to do this? Well, if we're talking about PCR, which isn't a cell, but if we're talking about PCR, what we do is we heat that DNA up and kind of melt it apart. Obviously, you can't do this because you can't just go put yourself in an oven and be like, all right, now I need to replicate my cells because you'll die. But what the cell does is it actually uses initiator proteins that are able to get in there and open up that DNA and get it ready for replication. And so these initiator proteins bind at sites in the genome called origin sites. And so for bacteria, this is a single site in the whole genome. However, for eukarya and archaea, there are multiple origin sites. And for instance, we have about 30,000 to 50,000 different origin sites in our genome. And this has a lot to do with the fact that our genomes are a bit more complicated because we have histone proteins and we got all of this wrapping up of genetic material. Also, we can fit inside these really small spaces, specifically the nucleus. And if I were to ask you, what do these origins tend to look like? You should start thinking about that. You should be thinking of an answer inside your head right now. That answer is that they tend to be rich in A's and T's because there's less hydrogen bonding between A's and T's, and therefore it's easier to open up the genome at those points. All right, so that is initiation. The next step in DNA replication is elongation. So this is the point in which both of these daughter strands get synthesized from the parental strand template. And there is a bunch of different proteins that help in this process. And I'm going to talk about what each one of them does and kind of the role they play in DNA replication. And my goal is to kind of present them in a somewhat chronological order of, this binds first and this happens and this comes along to kind of tell you the story of DNA replication. So you have these initiator proteins come in and they pull apart the DNA. And then you can have these proteins called helicase that come in and they further unzip the DNA. So you can imagine them kind of as the zipper on the zipper, if that makes sense. You grab your little zipper and start pulling on it, and that's what it does to the DNA. It slowly kind of pulls them apart upstream of where replication is happening. And it's important to note that this process requires energy. So you need ATP in order to break the hydrogen bonds between these different nitrogenous base pairs. And the opening up of the DNA in this manner causes a replication fork to form, which looks like the letter Y. So you have the splitting of these base paired strands to produce the head of the Y, and the tail of the Y is that double-stranded DNA. And so you end up forming 2 of these forks, one at kind of the tail end of the DNA, one at the head end, and they keep pushing outwards away from the origin of replication. And this forms what is called a replication bubble. So as I said before, DNA is very stable. So it tends to form these really strong hydrogen bonds, doesn't like to be pulled apart. For instance, as part of the research I do, I make a lot of single-stranded DNA in the form of complementary DNA. And we have to be really, really, really careful that when storing it, we use a special kind of buffer that makes sure that it doesn't stick to the walls of the tubes we put it in because it's just so sticky. So how exactly are these strands held apart when this bubble forms during DNA replication? Well, single-stranded binding proteins, or SSBPs, actually bind to the DNA and kind of hold the DNA open, shout out Hodor, to keep it from coming back together. Next, an enzyme called primase comes along and actually catalyzes the synthesis of these short RNA primer sequences that can then be recognized by the DNA polymerase. So basically, these are the same thing as primers in a PCR reaction, but this just helps the DNA polymerase bind and be able to start replicating the DNA. And the DNA polymerase actually recognizes the free 3' hydroxyl group that's on the RNA in order to bind and to start DNA replication. And so now we get to the star of the show, which is DNA polymerase. And DNA polymerase, as I said, is able to recognize the RNA primer and to bind to that and to start replicating the DNA. And so what it does is it recognizes, binds, and then starts extending that primer. And it adds on these DNTPs, which are deoxyribonucleotide triphosphates, basically just the building blocks of DNA. And it's really, really, really important to know the directionality of the polymerase. So the polymerase has 5' to 3' activity. In other words, it adds these DNTPs onto the 3' end of the growing DNA molecule. That's also to say then that the DNA polymerase travels from the 5' end of the DNA to the 3' end of the DNA as it's making that strand. The DNA polymerase also has what is known as 3' to 5' exonuclease activity. So in other words, it can actually go back in the opposite direction and proofread. And so this got me kind of thinking, is it that the same polymerase that just made this DNA goes back and like checks as it's going? Is it like, oh shit, I did it again, and then like goes and fixes? Or is it something like you got this boss polymerase that is kind of watching and, if one of them makes a mistake, it goes over and is like, oh, come on, man, there's wrong base, and then goes and fixes that and repairs that at that time. So, what is the mechanism here? And like so many things in biology, the answer is that a little bit of both of those happens during DNA replication. And another thing to mention about DNA polymerases is that there's a bunch of different kinds of DNA polymerases. And I'm going to talk about the differences between prokaryotic DNA replication and eukaryotic DNA replication. And I'll bring up the different types of DNA polymerases during that segment. And since the DNA polymerase has a single directionality in terms of its polymerase function of adding these bases, again, that is in the 5' to 3' direction, then you produce these two different strand types at the replication fork, one of which is the leading strand and one of which is the lagging strand. The leading strand is synthesized continuously by DNA polymerase, while the lagging strand is synthesized in these short little fragments known as Okazaki fragments. And so essentially you can imagine it like this. So you have this leading strand, synthesis goes towards the replication fork. So as this fork continues to open up, it's like riding a wave, You're just going to continuously keep going and going and going as long as that replication fork keeps opening up and opening up. On the other hand, you have these discontinuous fragments that are produced. So as you open up the replication fork a little bit further, then your polymerase can go up a little bit further in the DNA and start to replicate that, and it goes back down away from the replication fork. And then you have to do another one as it's opened up a bit more. Then as the DNA is opened up a bit more, you have another polymerase go up there and bind a little bit closer to that replication fork and go away until the fragment that is being produced reaches the fragment from before. And so that's kind of the difference between the leading strand, which is like riding a wave, and the lagging strand, which is made discontinuously. If you're having trouble picturing this in your head, just go look up a video or a picture or something. But I think visually, seeing this kind of helps you see what's going on. So yeah, if you're confused, go look up a video. So back to our proteins. So we got this DNA polymerase that is synthesizing these new DNA strands. Another important protein is topoisomerase. And so what topoisomerase does is it goes a little bit upstream of these replication forks and starts to cut DNA and kind of unwinds that DNA to relieve a lot of the torsional strain that is produced by opening up this replication fork. If you remember from biochem or even intro biology, the structure of DNA is this helical structure. So as you pull apart the individual strands that make up this DNA, then you start to put more and more torsional strain on the DNA that's downstream. So topoisomerase just comes in, cuts the DNA, kind of relieves that strain to make sure the DNA doesn't like blow up or, break under all that strain. So that is elongation. Let's talk now about termination of DNA replication. So one of the first things that's got to happen is you got to get rid of those RNA primers. So what happens is a enzyme called RNase comes in and chews up these RNA primers, and then a different kind of polymerase, it's still DNA polymerase, it's a different kind of DNA polymerase, and I'll talk about that in a sec, comes in and it kind of fills in the gaps of what was this RNA primer. Then you have an important protein called DNA ligase that comes in and facilitates the formation of phosphodiester bonds between nucleotides in the DNA. And so you can kind of think of this as a gap sealer. So it seals the gaps between these Okazaki fragments and between bases that have just been put in for the RNA primers. So it just kind of fills in these little gaps, little spaces that are in the newly replicated DNA strand. DNA ligase is also pretty important for DNA repair and then fixing bases that have been excised. So it plays a pretty big role in terms of sealing together bases that have just been put into the DNA. All right, so that is the process of terminating DNA replication. However, I want to kind of mention here what happens at the end of chromosomes in terms of replication. And so at the end of our linear chromosomes, we have these things called telomeres, and they are very repetitive in sequence. It's like TTAGGGG repeated 100 to 1,000 times, depending on the chromosome. And basically what they do is prevent valuable genetic information from being lost during replication. Because essentially what happens at the ends of chromosomes is that we have these RNA primers that are bound to the end of the chromosome and they get chewed up by that RNase enzyme, but then they are unable to be filled in by the DNA polymerase because it can't fill in at the end of the chromosomes. So then we lose that little bit of genetic information. So this telomere region basically acts as a buffer. And eventually what happens is that these telomeres get shortened and shortened and shortened until you actually do start losing some of that good genetic information. And this is over years and years, but eventually you do get to the point where you lose these telomeres. However, there's a really important enzyme called telomerase that is actually able to extend the telomere by laying down more of this telomere. You can think about it like laying down more track for a train. It's able to do that through adding a template, and then the DNA polymerase can come in and make the complementary strand to this template that is laid down by telomerase. So that's good, right? Telomerase comes in, makes the telomeres longer, and therefore we don't delete some of the important genes in the genome. However, a lot of the somatic cells in our bodies don't actually contain telomerase because telomerase is mostly found in germline cells. And so I think the thought used to be that we would be able to add telomerase to somatic cells, and this would drastically improve our lifespan, and it would make us healthier, live longer. And I don't think that's the case. I'm not updated on the most recent literature in terms of telomeres and lengthening telomeres in somatic cells and what effects that has. I know it plays a role in cancer. And this is a pretty interesting topic, and I would love to do a bonus episode on this, or, if someone out there is somewhat of an expert or does research in telomeres and would want to talk about it, I'd love to do an interview. Whatever it may be looking out for something in the future in terms of talking about telomeres, longevity, cancer, and topics surrounding that. All right, so that is a brief introduction into the way that DNA replication works in terms of initiation, elongation, and termination. The last thing I want to mention here is the difference in eukaryotic DNA replication and bacterial or prokaryotic replication. So there are three main differences that I'm going to talk about in terms of DNA replication in eukaryotes versus prokaryotes. So the first is in different types of polymerases. The second is in terms of initiation sites, which I mentioned, but I'll mention again. And then the third is in terms of termination. All right, so let's talk polymerases. So for bacteria, the polymerases are labeled with Roman numerals. So you have DNA polymerase 1, DNA polymerase 2, DNA polymerase 3. And there's more after that. I think there's 5, but the 3 are really the important ones to know. So in bacteria, DNA polymerase 3 is the main enzyme that adds nucleotides in the 5' to 3' direction and kind of serves that main function of a DNA polymerase. On the other hand, DNA polymerase 1 in bacteria has exonuclease activity and removes RNA primers and replaces them with newly synthesized DNA. So that has the exonuclease activity. And then polymerase 2 has a repair function, mostly functions through, again, that exonuclease activity. So big takeaway for bacteria is they have DNA polymerases that are labeled with Roman numerals, and there's kind of 3 important main ones. On the other hand, eukaryotic DNA polymerases are labeled with Greek letters. So there's a ton of different ones. I'm just going to present to you kind of the most important ones. So the first one here is the alpha DNA polymerase. And what alpha DNA polymerase does is it synthesizes the RNA primers in order to initiate DNA replication. So it does this in the leading strand and also the lagging strand. So alpha polymerase plays a role in initiating DNA replication. Next, we have beta DNA polymerase, which plays a role in repairing DNA. The next is gamma DNA polymerase, and that plays a role in replicating mitochondrial DNA. Mitochondrial DNA is important because it is inherited from your mother, so a lot of these genetic diseases that are passed down maternally are due to inheritance through mitochondrial DNA. The next DNA polymerase is called the delta DNA polymerase, and this is kind of the star of the show. This plays the biggest role in DNA replication in eukaryotes. So the delta DNA polymerase synthesizes both the leading and the lagging strand and fills in the gaps after the removal of the primers by the RNase enzyme. The last polymerase you need to know is the epsilon DNA polymerase, and its role is in repairing DNA. So I think the main takeaway here is to remember 2 eukaryotic DNA polymerases. That is the alpha DNA polymerase and the delta DNA polymerase. So the alpha's role is to come in early, set down these RNA primers, so then the delta DNA polymerase can come bind to those primers and then start synthesizing the leading and the lagging strand. And then that delta polymerase can also come in afterwards and fill in the gaps that are left once these primers are removed. All right, so that is a little bit about DNA replication. I think the really main takeaways here are, number 1, the location in which DNA replication occurs, that is in the nucleus, since that is where the DNA is. And then you need to remember the directionality that these enzymes go. So you got to remember that the DNA polymerase goes in the 5' to 3' direction. That is, it adds these DNTPs or deoxyribonucleotide triphosphates onto the 3' end of the growing DNA strand. And I think it's important to remember that this directionality is in terms of synthesis. So this is the direction the enzyme goes in reference to this daughter strand. If you're looking at the parent strand, then you would say the enzyme travels in the 3' to 5' direction. So that's just something to think about because, the MCAT might try to be a little tricky and say, what direction does the DNA polymerase travel in terms of the template strand or in terms of the coding strand? In which case you would have to know that it travels the opposite direction, which again, 3' to 5', because these DNA strands are anti-parallel. So you might want to put this on your note card, but synthesis is in the 5' to 3' direction. In terms of the template strand, the DNA polymerase travels in the 3' to 5' direction.
Thanks for checking out the Prospective Doctor MCAT Basics podcast. Sam's doing a killer job taking you through the most important MCAT topics, but what if you need a little extra help? How does a 5, 10, or even 15-point increase in your score sound? Imagine how your chances at admission could increase. Med School Coach's MCAT tutoring can get you there. With the most rigorous selection process of any tutoring company, we see amazing results. We deconstruct each student, find a plan that is going to work, and help execute it. That's why our students add an average 12 points to their score. Completely physician-run and operated, and focusing on nothing but medicine. It's no wonder over 10,000 past students have trusted Med School Coach to get them through the MCAT and into medical school. Check out medschoolcoach.com today and mention code PODCAST for 5% off.
All right, so the next topic I want to talk about is central dogma. So central dogma is the process in which DNA is converted to messenger RNA, which is then converted to protein. This term was first coined by Francis Crick, who helped determine the structure of DNA. And this is broken down into two processes, the first of which is the conversion of DNA to messenger RNA, which is known as transcription. The second is the conversion of mRNA into protein, which is known as translation. So let's talk about the first of those two processes, transcription. So transcription occurs in the nucleus and has three stages, which is very similar to replication. It has initiation, elongation, and termination. So in terms of initiation, the first thing that has to occur is RNA polymerase, which is a little bit like DNA polymerase, has to bind to the DNA at the promoter. And RNA polymerase needs a little bit of help to do this. So in bacteria, there is a protein called sigma factor that actually must associate with the polymerase and bind to it before it's able to bind to that DNA promoter. In eukaryotic cells, it's a bit more complicated. You got to have a lot of these different factors bind to the RNA polymerase in order for it to associate with the promoter region of a gene. So just know that for the prokaryotes, you have the sigma factor. For eukaryotes, it's a lot more complex and, it's over the scope of the MCAT. So the promoter region of a gene is this little region before the gene or upstream of a gene in which the RNA polymerase recognizes and then can start transcription. So there's a few different pretty well-characterized promoters. The first is found in archaea and eukarya and known as the Tata box, which is not to be confused with the Toodaloo box. That's a different one. And then in bacteria, the most highly characterized promoter region, which is kind of analogous to the Tata box, is the Pribnow box. And before someone gets mad at me, I just want to say that it's definitely not limited to these two kind of well-characterized promoters. There's a lot of different kinds of promoters. If you're really interested, go watch a YouTube video or something like that. But the most well-known, most likely to see on the MCAT is the number one, the Tata box, which is in archaea and eukarya, and then the Pribnow box, which is found in bacteria. All right, so back to initiation. So what's happened so far is that we've had this RNA polymerase associate with some other factors, sigma factor if it's a bacteria, and it has found the promoter region and it's bound to the promoter region. What happens next? Well, now we start into elongation. So when this RNA polymerase binds to DNA, it undergoes a conformational change from the closed conformation to the open conformation. And this opens up the DNA duplex, kind of similar to what some of these initiator proteins do in DNA replication. And so they open up that DNA duplex and they create what's called a transcription bubble. And this transcription bubble is about 13 base pairs long. So you open up this little bubble and then now you can start transcribing this DNA into messenger RNA. And so as this RNA polymerase travels along the template strand, it starts to produce an RNA strand that is complementary to the template strand. So the strand essentially being used by the RNA polymerase is called the template strand. The other strand that's not being used by the RNA polymerase is called the coding strand. And the coding strand is going to have the same exact sequence as the RNA strand being produced, except of course you'll have uracil instead of thymine because that's what RNA has instead of DNA. But that's important to know is that the coding strand of DNA will actually have the same exact sequence as the RNA strand that's being produced by RNA polymerase. And similarly to DNA replication, the directionality of the RNA polymerase is in the 5' to 3' direction. That is, RNA bases are added on to the 3' end of this mRNA as the transcript grows. All right, so now we have RNA polymerase traveling along this gene, and it's turning this gene from DNA into RNA. And what happens when this gets to the end of the gene, or how does this get to the end of the gene? How does it know when to stop producing this messenger RNA? Well, we have studied a few of these mechanisms, and we know this process is called termination, and it happens differently in prokaryotes versus eukaryotes. So for prokaryotes, there's a few different ways this happens. The first is that this RNA can form a hairpin. You have base pairing between different parts of this transcript that actually cause the transcript to form kind of this loop structure, which in turn causes the RNA polymerase to essentially fall off of the DNA and quit transcription. The next mechanism that prokaryotes use in order to terminate transcription is using this Rho factor protein. And so what the Rho factor protein does is it binds to the DNA RNA complex that has formed from transcription, and it actually unwinds it. And it uses ATP to do this, and it kind of follows behind the polymerase. I imagine it a little bit like a shark kind of swimming behind this polymerase and using its fin to just like tink, 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 like break up these hydrogen bonds between the RNA and DNA. And then once the polymerase finishes, this transcript is basically freed because you have this Rho factor shark just with its fin popping off this RNA. And then, of course, the shark eats the polymerase. So again, for prokaryotes, the RNA can form a hairpin, and then that can kind of kick the RNA polymerase off. Or you can have this mechanism in which the Rho factor kind of follows behind this polymerase and basically unwinds the RNA DNA complex and terminates transcription. In terms of termination in eukaryotes, I wouldn't worry too much about understanding the different processes that happen. I don't even understand it. I think all I know is it's really complicated, and I know I've said this, but it is outside the scope of the MCAT. Instead, I'd focus on some of the post-transcriptional modifications that occur in eukaryotes. That is the addition of a poly A tail on the 3' end of the transcript, addition of a 5' guanosine cap, and then also intron-exon splicing, which basically removes bits of the RNA transcripts and then splices together all the remaining bits. So I think that's really important stuff to understand, this transcript processing that occurs in eukaryotes. So let's take a little bit of a deeper dive into transcript processing. So as I said, there are 3 main post-transcriptional modifications that happen to eukaryotic mRNA. And just a little side note, transcribed RNA that has not undergone these modifications is considered pre-mRNA, or hnRNA, which stands for heterogeneous nuclear RNA. I like to think of this abbreviation as has not, as in has not reached puberty or has not reached maturation. All right, so let's break down these 3 modifications. The first modification to be added is a 5' cap, and it is a modified guanosine or modified G base, which is a 7-methylguanosine, and that is added to the very first nucleotide in the transcript. And typically this is done before the transcript is even finished being transcribed, and therefore it is the first modification that occurs. And this modification protects the transcript from degradation and helps this transcript get recognized by the ribosome, which is important for the next step known as translation. And this modification occurs in the nucleus. The second modification is the addition of a poly A tail to the 3' end of the transcript. So the poly A is just a bunch of adenosine monophosphates that are added on to the transcript. So you'll just see A at the end of this transcript. And that protects against degradation. And this modification also occurs in the nucleus. And kind of interesting, one of the newer technologies in terms of research is being able to do something called RNA-seq, which basically looks at all of the genes that are being expressed in a cell. And then from there, you can do a lot of different things. So you can look at, maybe a particular cell type has this transcriptional profile, and maybe we can target, one of these upregulated genes to cure disease, whatever. And this RNA-seq method actually relies on this poly-A tail, because the first step and the first thing you need to do if you want to sequence all of the transcripts within a cell is you need to be able to isolate all of these messenger RNAs. And what is one characteristic of all messenger RNAs? Well, they all have poly-A tails. So what you do is you use some kind of primer that will actually bind to that RNA tail. So for instance, I have these beads that have what's known as oligo-DT conjugated to them. So it's basically this primer sequence. It's a bunch of T's, and you mix it with your sample, which most of which is actually ribosomal RNA. Something like 90-95% of all the RNA inside a cell is ribosomal, and then like 5 to 10% is mRNA. And so you mix these beads in, and the beads will actually bind all these messenger RNAs based on their poly-A tails, and then from there you can purify that messenger RNA and sequence it. And the third and final modification is RNA splicing. So what happens is you have the spliceosome that comes along, and it chops out introns, and that leaves the exons. So your final transcript will be all the exons put together and all the introns removed, which sounds backwards. I get that. It's very confusing. When I first took genetics, I was like, what the hell? Who named these? Kind of dumb, but introns are removed. They don't stay in. Exons, on the other hand, are kept in the mature transcript. And it's important to mention that different bits of the pre-mRNA can be removed in order to make different transcripts. And this is called alternative splicing. So, you could take bit 1 and link it to bit 3, and that's your mature transcript. Or you could take bit 2 and link that to bit 3, and now you have a different transcript. So this just gives our bodies or our cells even more control over gene expression. And like the other modifications, this occurs within the nucleus. And before I move on to translation, I want to briefly mention kind of the main differences between eukaryotic and prokaryotic transcription. So eukaryotic transcription has a lot more detail, it's a lot more complex. So you have transcript processing, and there's just a lot more going on at these different steps in general. And then for prokaryotic cells, there is no transcript processing, and in most cases, your mRNA is actually being transcribed and translated at the same exact time, which, I would hesitate to say that the mRNA is kind of floozy, but it's a little bit freaky. Another interesting thing in prokaryotes is that a lot of genes are organized into operons. And if you really want to know more about operons, go listen to my genetics podcast. You'll definitely see that come up. But essentially, a bunch of very similar genes are being driven all by the same promoter. All right, so where are we at? So we're at the point in which RNA polymerase has bound to the DNA. It's undergone a conformational change that kind of opens up the DNA, and then the mRNA has been transcribed, and that reaction has been terminated. And now you have this mRNA that, if you're a eukaryotic cell, has undergone these post-transcriptional modifications, and now it's floating out of the nucleus into the cytosol, where there's a bunch of ribosomes, and now we get into translation. All right, so let's talk about translation. So translation is the conversion of mRNA to protein, and this is done by ribosomes. And so very similarly to transcription and also DNA replication, the transcript is read in the 5' to 3' direction by the ribosome. And I remember this by thinking about the 5' cap that's put on mRNAs. And so that cap helps the ribosome recognize the mRNA. And so you can think, okay, that's the part that first enters the ribosome, and so then it must be read then from that 5' cap to the poly-A tail on the 3' end of the transcript. So let's briefly talk about ribosomes. So ribosomes are made up of ribosomal RNA and protein, and they have a small subunit and a large subunit, and they are very, very abundant in eukaryotic cells. There's something like 10 million ribosomes in each active eukaryotic cell. So they're very, very abundant. And then, as I talked about in the previous podcasts, prokaryotic cells have 70S ribosomes, and eukaryotic cells have 80S ribosomes. And there's 3 different sites of interest within the ribosome. There is the A site, which is the amino-acyl site, and I like to think of that as the acceptor site. And then there's the P site, which is the peptidyl site. And then there is the E site, which is the exit site. And I'm going to talk more about this in a minute, but just understand that these are kind of the three important sites within the ribosome that have to do with translation. And what it comes down to is this is three different sites where tRNAs bind. All right, so let's get into the three stages of translation. And I'm going to sound like a broken record here, but there are three different stages: initiation, elongation, termination. So the first step here is initiation. How exactly is translation initiated in the ribosome? Well, the first thing you got to know is that when the ribosome and the mRNA come together, it's kind of interesting because both the subunits come together and kind of sandwich the mRNA between them. So you can kind of think of it as a little bit of a ribosome mRNA sandwich. In terms of initiation, it's also important to understand how a ribosome recognizes the mRNA. So this is different for prokaryotes versus eukaryotes. So let's start with prokaryotes. For prokaryotic ribosomes, there's one thing that they recognize, and that is the Shine-Delgarno sequence. And this is typically found in the actual mRNA itself, and it's generally located about 8 bases upstream of the start codon, which is AUG. And of course, for eukaryotes, this process is a lot more complex, requires like 10 different proteins. But one thing to kind of remember is that the ribosome can recognize the 5' guanosine cap that is added as a post-transcriptional modification. I liken this to me skiing as a little kid. So, growing up in Colorado, I skied quite a bit, but as a really little kid, my parents loved to dress me up in this like neon yellow ski suit, from head to toe. I was neon yellow, and that was so that they could see me anywhere on the ski slope. Anywhere I went, they'd be like, okay, there's Sam, there he is running into a tree, whatever it is. They could always find me. In the same way, this 5' guanosine cap helps those ribosomes say, okay, there is an mRNA. Quickly, let's translate that into a protein so it can be used within the cell. All right, so now we're at the point in which we have this mRNA sandwiched between the large and small subunits of the ribosome. And the next step in initiation is that a tRNA, which is a transfer RNA that binds to the start codon, which is AUG, brings the amino acid methionine with it and binds to that AUG codon in the A site in the ribosome. And if you're like, hey man, what the hell? I don't know what a tRNA is. I don't know what a transfer RNA is. I haven't taken biochem in like 4 years. Well, let me quickly review this for you. So basically, a tRNA is this cross-shaped structure made out of RNA, and at one end, it's got this amino acid attached to it, and then at the other end, it has this anticodon loop. And this anticodon loop is made up of 3 bases, RNA bases, that bind to complementary bits of the mRNA. So, if there's 3 bases that are complementary to this tRNA on the mRNA, then they will bind, and that is what actually happens in these different sites. So let's talk about how this works. So for the start codon, which is AUG, the complementary sequence to that is UAC. So this tRNA would have an anticodon loop that contained UAC, and on the other end was attached to the amino acid methionine. So that's kind of what that looks like. All right, so now your ribosome's kind of all set up. It's got this methionine tRNA set up in its A site, and it's ready to start elongation. And so once this start codon tRNA is in the A site, the ribosome actually moves towards the 3' end of the mRNA. And what that does is that opens up the A site to new codon. So 3 more of these bases are now exposed in the A site, and now a new tRNA can come down, and if its anticodon loop is complementary to these new bases, then it stays, and now you have this new tRNA in the A site, and you have this other tRNA that's been moved to the P site. All right, so now you have a tRNA in the P site and a tRNA in the A site. And what happens is a peptide bond forms between the amino acids that are attached to these tRNA. And basically, the amino acid from the P site, or chain of amino acids in the P site, are transferred over to the tRNA that is in the A site. So you can imagine if there is just this methionine that is in the P site because this is only the second codon in the mRNA, then this methionine forms a peptide bond with whatever the amino acid is that is in the A site, and then is transferred onto that tRNA. So now you have this tRNA that's in the A site, and it's connected to one amino acid and then connected to methionine. So now you have this 2 amino acid peptide that is bound to a tRNA in the A site. And at this point, the ribosome moves down another codon or 3 letters, and the tRNA that's connected to these 2 amino acids is pushed into the P site. And what this does is then this pushes the naked tRNA, or the tRNA that just transferred that methionine over, to the E site. And at this point, that naked tRNA can kind of just float away off into the ether or whatever. But as I said, this exposes a new codon in the A site. So at this point, you can have a tRNA come in, and if its anti-codon loop is complementary to that codon, then you get binding, and then you form a peptide bond between the amino acid on that new tRNA and the peptide that's growing on the tRNA that's in the P site. And what this does is it essentially transfers the peptide over to the A site, and now you have this 3 amino acid peptide connected to the tRNA in the A site of the ribosome. And then again, your ribosome moves over, kicking that naked tRNA into the E site where it goes away. And then the tRNA that contains this 3 amino acid peptide is moved to the P site, and you expose this new codon. In comes another tRNA. You form another peptide bond, and those amino acids are transferred over to that tRNA in the A site, and then it moves over again. And then this cycle just repeats and repeats until you have formed your full polypeptide. And at this point, the only thing left to do is terminate translation. So translation is terminated when the ribosome encounters a stop codon. There's a few different stop codons. There is UAA, UAG, UGA. And basically what happens is that this stop codon is exposed in the A site, and all these tRNA together are all like, man, what the is this? They can't recognize it. They go in, they're like, okay, I don't match up, you don't match up. The tRNA cannot recognize a stop codon. So instead what happens is release factor proteins go in the ribosome and actually bind to the stop codon in the A site. And what this does is this triggers hydrolysis of the bond between the final tRNA and the peptide, and then that peptide is released into the cytosol. All right, so that's kind of a brief crash course into translation. The last thing I want to mention here is that, like post-transcriptional modifications, there can also be post-translational modifications. And I'm sure you've heard of many of these, and maybe you don't really think of them as post-translational modifications. So the first is phosphorylation. So proteins can be phosphorylated. That tends to activate them if they're phosphorylated, or deactivate them if they're dephosphorylated. So in a way, the cell can kind of like add batteries to the protein, turn it on and off, or flip a switch on the protein, however you want to think about it. Proteins can also be carboxylated, and this is adding a carboxylic acid group to the protein. And this is primarily done in proteins that are involved in blood clotting. The next type of post-translational modification is called glycosylation, and this is simply adding a carbohydrate group to a protein. And what this tends to do is mark proteins for particular cellular destinations. So say, you want to exocytose this protein you just made. Okay, well, I'll mark it with this certain carbohydrate, and the cell will know that this protein needs to leave. Or, maybe I'm trying to target a protein to the Golgi apparatus or whatever it is. Glycosylation tends to mark proteins for certain destinations. It also plays an important role in cell-to-cell adhesion, so that's something else to remember. The last post-translational modification I'm going to talk about is called parental or lipidation, and that is the addition of a hydrophobic group to a protein. And again, this plays a role in determining cellular destination of a protein. So what I wanted to have you thinking here is, after a protein's made, I can do a bunch of different stuff to it. I can add fatty groups, I can add carbohydrates, I can add phosphoryl groups. You can do a lot to a protein after it has been translated. All right, and that is it for the Central Dogma and DNA Replication podcast. Thank you for listening as always. If you liked the content in the podcast or you feel like this helped you study for the MCAT, please go give us a review on iTunes. Helps us in the rankings and helps us reach more people. I'm coming out with a bonus episode with an MCAT tutor from Med School Coach, whose name is Catherine. She also runs the Pre-Med Instagram, and super smart. It's a great interview. So look for that at the end of this week, early next week. It's going to be a great interview. Lots of advice about studying for the MCAT in general. A lot of stuff about taking a gap year, as both her and I are taking gap years. Kind of, what to focus on in a gap year, how to help your application and everything. And also a bit of advice about gaming the Instagram algorithm system. So once that comes out, take a listen. Thanks as always for listening, and I'll see you for the next one.
Thanks for listening to Prospective Doctor's MCAT podcast. If you're a pre-med, you'll want to check out prospectivedoctor.com for tons of free tools, articles, and more podcasts that cover your pre-med life. And if you need help on the MCAT or getting into med school, check out medschoolcoach.com for the experts.